Hello everyone, this is Manjusha Dixit from National Institute of Science Education and Research, India. In the background, you can see my institute's campus. So today I'm going to talk about the role of eukaryotic elongation factor 1A2 in triggering ERK-mediated metastatic program in ER-negative versus ER-positive cells. So this work has been published in Life Sciences in 2020. So eukaryotic elongation factor 1A is the most abundant protein. Its role is to deliver amino acyl tRNA during the elongation step. Here you can see that at ribosome it delivers the amino acyl tRNA. But in addition to its canonical role, several moonlighting functions have been reported, which include GDP exchange, nuclear export, protein degradation, actin bundling, apoptosis, viral functions. So some interesting point regarding this eukaryotic elongation factor 1A is it has two isoforms, 1A1 and 1A2. I'll be referring them as in short as 1A1 and 1A2. So 1A1 and 1A2, they show 92% identity at DNA level, and each one of them has an independent gene. For 1A1, it is present at chromosome 6, and for 1A2, it is present on chromosome 20. So the interesting point is, even after this much identity, whatever differences are present between these two. So this publication in PLOS ONE shows that they are present on the surface and they are clustered. These two clusters you can see. So because of these differences, each isoform shows different non-canonical functions. And regarding their expression, it has been shown that 1A1 is ubiquitously expressed, means most of the organs you will find the expression of 1A1 isoform. But in some organs, like in brain, heart, skeletal muscle, 1A2 isoform is reported. So actually what happens, this mouse-based study, it showed that 7 days post-birth, this 1A2 replaces 1A1. And by day 20, it is completely replaced. So it's not like these organs are having the expression of both the isoforms. It is only isoform 1A2 is expressed. And this 1A2 isoform is indispensable for the survival. This study reported that mutation in 1A2 was lethal. So that shows that when this switching happens, switching of isoform happens, so 1A2 isoform is, it takes over completely the function of 1A1 isoform. But in cancers, the situation is different. There are several studies. This one, first time it was reported in ovarian cancer, as the expression of 1A2 was increased compared to the normal tissue. And then a study in breast cancer, and this is another study in ovarian cancer, this is study in prostate cancer, and more studies have come which show that the expression of 1A2 is increased in these tumor samples compared to the normal samples. So this developmental switch we discussed in previous slides, where in certain organs 1A2 takes over completely the function of 1A1. But in case of tumors, 1A1 is expressed, this isoform is expressed, but in addition to 1A1, the isoform 1A2 is also switched on. So based upon this, we hypothesize or we consider that it can be oncogenic switch. So the expression, increase in the expression in 1A2 may give tumorigenic properties to the cells. So what's its connection with survival? Several contradictory findings have been reported. So in this ovarian cancer and versus non-small cell lung cancer, here especially if you notice that the increased expression was associated with the better survivability. So generally for oncogene, this is unique observation that generally they are associated with the poor survivability. So here the just opposite was reported. And if you look at the breast cancers, this study showed that the high 1A2 expression was associated with better probability of the survival. And in this study, they also checked that what is the association. It had something to do with the ER/PR HER2 status or not. So they found that this high expression of 1A2, it predicted good prognosis in subset of the patient samples with lymph node positivity and HER2 negative tumors. So just this was an interesting observation, but it was not consistent. In another study in triple negative breast cancer samples, that increased 1A2 expression was associated with the poor prognosis. So overall, based upon these studies, it can be considered that it might have something to do with the ER receptor positivity, but its correlation or its connection with ER receptor is not properly understood. For that also there are contradictory reports. So based upon that, we hypothesize that that 1A2 may give good prognosis due to its role in metastatic program. So there might be difference in the metastatic program, which is triggered in ER positive versus ER negative cells. There might be crosstalk between 1A2 mediated signaling versus ER mediated signaling, or somehow they can have crosstalk, and it may alter the metastatic program, which is triggered in ER positive versus ER negative breast cancers. So this same has been shown in this publication also, that there might be difference in the aggressiveness across the molecular subtypes of the cancers. So we started this work by using TCGA based data. So if you see here that when we divided samples in primary tumor versus metastatic tumors, so between primary and metastatic tumors, there was no significant difference in 1A2 expression levels. But if you compare with the normal, yes, it was in tumors, the expression was higher. When we divided the samples based upon their molecular subtypes, based upon the SPAM50 subtypes, so we found that across these molecular subtypes, if you compare the 1A2 expression levels, there was difference in certain cancer types. So this data is for 1A2, just as control that how 1A1 was behaving, we checked for 1A1 also, but in 1A1 we did not find any difference across the subtypes, especially here if you see that there was no significant difference in luminal A versus basal type. Then we, just to check that how it was interacting with ER receptor, was there any difference in ER positive versus ER negative samples? So indeed, we found that there was difference in ER positive. The expression level of 1A2 was higher compared to the ER negative samples. And it very well here it shows that ESR1 is the gene for ER. So the expression levels of 1A2 very well correlate with the expression levels of ESR in this TCGA data. And if you see for 1A1, where there was no significant difference. So based upon that, we concluded that the expression levels of 1A2 might be different in ER positive versus ER negative samples. So to check that how 1A2 and ER are affecting the expression levels of each other, there might be any feedback loop and which direction this loop is going. So first of all, we took multiple types of breast cancer cell lines. Here their molecular subtypes are written here. So if you see that this cell line, which is ER negative, and this is also ER negative, this is also ER negative, these three. So if you compare with these two, which are ER positive lines, the expression was reduced. Here also it is not as less as these two, but still compared to these ones, it was lesser. So this is the densitometric data for the same. And at mRNA level also we made similar type of observation. So just to check that how 1A2 was affecting the ER expression levels, we prepared the stable lines, first of all in MCF7, which is ER positive line. So we depleted the expression of 1A2 in this stable line, and we checked that what was the effect on ESR1 expression, and we found that it was reduced in this knockdown line. So that showed that 1A2 expression was affecting ESR1 expression levels. So to further check that was it true other way also, what was ER affecting the expression level of 1A2? So we treated the cells with ER antagonist tamoxifen at different time points, and there we found that after this is the control. So compared to control, the expression of 1A2 was decreased, and PS2 is a known target for ER, so that showed our system was working all right. So based upon that, we conclude that there was a positive feedback loop where ER was affecting the expression level of 1A2 and 1A2 was affecting the expression level of ER. So to further check that was there any difference in the metastatic properties between ER positive versus ER negative cells, we did these migration based assays. So this data is from MCF7 cell, which is ER positive. So here, if you see that this is wound healing assay, this is transfill migration assay. So we did not find any significant difference between the 1A2 depleted cells versus the control. And we checked the markers for EMT also. So these markers also did not show any dramatic difference between these two groups. And MMPs play important role during metastasis. So these MMPs were also not altered significantly. Then we tried other way also. We increased the expression level of 1A2 and MCF7 cell line. And there also for the wound healing and transfer migration, we did not find any significant difference between the two groups. And at molecular level, N-cadherin was showing significant difference, but E-cadherin was not showing. Vimentin was showing, but here MMP1 was not showing. So again, based upon this, we concluded that it might be affecting the metastasis. The expression level of 1A2 might be affecting metastasis, but we did not see any consistent or dramatic changes. So in next part of the work, we choose triple negative cell line, that is MBMB231, and we checked all these properties and checked that what was the level of trigger of metastatic program. And there we found this is wound healing assay and this is transfer. Both of them, you can see that clearly there was dramatic difference between these two groups, because endogenous levels of 1 or 2 are very less in these triple negative lines. So we just worked with the ectopic expression based system. So both of them, they are showing clear increase in the migration rate here. So at molecular level also, we could see that clearly that these all EMT markers, their expression was increased with the increase in 1A2 expression level. And though phospho AKT levels were not changed significantly, but there was clear-cut activation of ERK signaling. So based upon that, we concluded that this trigger of EMT program might be ERK mediated. So to further confirm that, we inhibited ERK on 1A2 overexpression background. And by this inhibition, we could see the rescue of the phenotype. So if you compare, this is equivalent to the control group. So this suggested that yes, this metastasis was ERK mediated. Similar kind of rescue was observed in this transwell migration assay also. So these two graphs are showing the quantitative figures that you can see the control values and overexpression with ERK inhibition. These values are equivalent in both the cases. So this suggested that migration was ERK mediated. So to further look into the molecular mechanism, that this CXCL1, 8 and CXCR2 axis is well reported to increase the as downstream target of the ERK. So we checked the expression levels of CXCL1 and 8 by using RT-PCR and ELISA. Both the data sets, they showed that there was significant increase in these two. Okay, so to confirm this further, on ERK inhibition background, we checked how are the levels of CXCL8. So again, we see that there was rescue. So that showed that yes, CXCL was downstream of ERK. And same this migration assay, here we inhibited this CXCR2 receptor, and with the inhibition of this receptor, we could see the rescue of the phenotype in this overexpression, ectopic expression background. By inhibition, we can see that there was rescue in the phenotype. So compared to this one, now this is equivalent to the control. So this showed that that 1A2 expression was affecting ERK, and ERK was affecting the expression levels of CXCL8 and CXCL1, and these were binding to CXCR2 receptor, and they were responsible for the phenotypes we observed. So overall, we conclude in this study that 1A2 is preferentially expressed in tumors and cancer lines of luminal origin, and it regulates ER levels in MCF7 cell lines, which can explain its association with the good prognosis in breast cancer. And this 1A2 induces EMT in triple negative cell lines. We are activating ERK pathway, and definitely if we compare in ER positive versus ER negative cell lines, the robustness of the metastatic program, the trigger is much more pronounced in triple negative cell lines. So this all work was done by my graduate student, Khurshidul Hassan, who is now postdoc. And this is my research team. And thanks to my funding agencies, DAE and my institute, NICER. Thank you very much. I can take the questions now.